It's my great pleasure to be here. Thank you very much. The microbiology of the built environment is an unusual ecosystem for anybody to play in, especially for an ecologist. But then I am a microbial ecologist who became a professor of surgery, so nothing really has to make sense anymore. We tend to explore how bacterial communities work and how they interact with each other and with their environment. And it's irrelevant, completely irrelevant, where those ecosystems are, whether they're on your jacket, right? You know, you have a microbial ecology on your jacket. No matter how much you steam it, they're still there, right? There's a microbial ecosystem in your intestine, right? There's a microbial ecosystem on your chair. There's a microbial ecosystem in the grass outside and in the plants. And even on these windows, there are cyanobacteria happily growing and metabolizing and producing energy and replicating. That's microbes, right? There's 3.8 billion years of genetic evolution to reach a peak of what we would consider the optimal life. These are the ultimate living beings on the planet, right? Completely and utterly changing the face of this ecosystem and the world we live in. Now, we've created this new world, and they are adapting to it. They are evolutionarily adapting to it. They are changing their metabolism. They're changing how they interact with each other, because this ecosystem is actually quite alien to anything else they've ever had to deal with, okay? It's very dry. It's got a lot of oxygen. It's generally quite benign in terms of the surface structure. It has limited niche space. And so all of those things which humans inadvertently did to make it more comfortable for themselves make it actually very inhospitable to the microbes. And that inhospitability, is that a word? I made it up, it's a good word. The inhospitability of the built environment is exactly what makes this ecosystem so interesting to microbial ecologists, physiologists, evolutionary biologists, because that inhospitality drives these organisms to adapt, to become more dependent upon themselves and each other to create new ecosystem dynamics. The human microbiome is the predominant source of bacteria in the built environment. We know this because when we go into all of our built environments, we find that the vast majority of bacteria come from the people, the occupants in that space. Your human microbiome is some 40 trillion bacterial cells, around 9 million genes, which makes your paltry 25,000 genes look a little bit irrelevant now. And they adapted to your ecosystem as well as your ecosystem adapting to them. Your immune system, for example, is just an extension of our body's ability to manage the microbiome. We like to think of the immune system as a national park warden, right? Or a gardener, going out there, removing the weeds, making the good things grow, keeping the things that are beneficial around, and shaping that ecosystem in a way that's most effective for the health of the person, okay? And so this ecosystem interaction has led to a unique microbial profile for individuals. We get that microbial profile predominantly from our mother, so it's handed down to us from generation to generation. So you don't just get the genetic information and a lot of interesting and useful facts from your mother, you also get a huge microbial burden. Vast majority coming when you're born. From a vaginal delivery, you get a large amount of microbes from your mother's vaginal cavity. If you're born by a C-section, you get microbes from your mother's skin, right? But then that changes as you develop. Within the first few months of life, the microbiome of an infant usually looks quite similar, right? All infants, no matter how they're born, by the age of 3 months, have a very standard-looking microbiome. It's very independently different between the infants, and it varies day by day, but there's no real difference associated with birth mode. So where do those bacteria come from? Where do those fungi come from? Where do the viruses come from that populate those children? Paula was mentioning a study in a neonatal intensive care unit where they found that some of the bacteria from the gunk in the sink were colonizing the kids, right? But what if it was from the animals as well that you interact with? What if it was from the land you went out and walked on or interacted with? What if those very experiences were the ones that shaped your ecosystem most effectively? And we're interested in that because that leads to a unique signature. And that unique signature is important for many different areas of our understanding of medicine and health. But it also can help us in unique systems like forensics, and I'll talk a little bit about that. I call this talk forensic microbiology because we're using techniques now to not just use microbial ecosystem dynamics as a forensic tool for say, criminal activity, but we're also using it to try and understand and shape our appreciation of how these ecosystems vary. Okay, so for precision medicine, we still apply a forensic approach. I want to determine forensically whether my patient is going to survive if they go through this particular procedure. I want to determine forensically if this particular medical therapy is going to be effective for my particular patient. And so that's a forensic application too. And so we're really interested in applying forensic tools to understand the microbiome in myriad environments for myriad purposes. So we think that our lives have been changing over the last little while, from playing outside where we have a diverse microbiome to living and playing inside where we have a limited microbial exposure. We've actually interrupted our environment, shaping it to kill the good bacteria along with the bad, and leaving ourselves open to the development of atopy and allergies and asthma. And this is potentially shifting our lifestyle choices, right? We think that by promoting a microbial ecosystem inside us, maybe adding probiotics, we can actually protect our children and maybe our adults from developing some of the autoimmune conditions which they have become so prevalent in our society. You know, the food allergies, the skin disorders, the major autoimmune conditions, even the metabolic conditions like diabetes and obesity. We think we can alter the trajectory of those. And there's two ways of doing that. One is by fermenting or altering the microbiome inside our bodies. And the other one is by altering that microbial exposure. And so we've done this focusing on many different groups of people. For example, the Amish and Hutterite communities that live up in North Dakota and Indiana. This is an Indianan Amish farm. Anyone who doesn't know what the Amish are, they're a sect from Eastern Europe, Central Europe, and they have a very agrarian lifestyle. They eschew technologies. They don't have any iPads. They don't have any telephones or video screens, no electricity in the home. The homes are very, very clean. The Amish live on personal family farms, and all the children muck in on the farm. They work there from very early in life. They're very industrious. They're driven to have an industrious lifestyle with no frivolity. But the most interesting thing, that lack of iPads. So I can take my kids down to this Amish farm in northern Indiana and show them that children can survive without an iPad. Right? It's technically possible to live. That environment we think is highly protective for these children. If we look at the Hutterite community, very similar lifestyle choices. They live the same kind of ecosystem, right? They have no technology, they have no iPads, no screens, no electricity in the home. But they've elected to live on big communal farms. And they've also elected to make their farms highly mechanized, which has certain health and safety implications, so the children are not allowed on the farm, okay? And both the Amish and the Hutterites have the same genetic background. They have exactly the same genetic markers for asthma, okay? And they came over here from very similar backgrounds, from both Sweden and Switzerland, but from similar backgrounds in Eastern Europe, migrated to the States, adopted these two lifestyle choices, which at home are identical, but in their farming environment are very different. And the Amish, well, they have about 4% asthma in their population. You know, a very small percentage of the children develop asthma. The average for the US is about 9%. But the average for the Hutterite population is around 35%. So 35% of the children in the Hutterite community are developing asthma. Why? We think it's because of that interruption. And we can see that those children are not allowed to interact with the farming environment, and it shapes how their built environment in their homes offers a protective medium. And what I mean, I'll show you exactly what I mean by that. Number one, the children have very different immune profiles. So you take a child from the Amish population, they'll have if you look at the peripheral blood leukocytes, they'll have an increased proportion of neutrophils, but most importantly of all, those neutrophils are less aged. And by that I mean they're in circulation for a shorter period of time in the body. They're rejuvenated more frequently from the bone marrow. And that rejuvenation leads to a reduction in the inflammatory potential of that system, right? So something about the environment in which these kids live is reducing the number of aged neutrophils in the population of their blood, and that reduces the likelihood that they're going to have an inflammatory event. The inflammatory processes that inflammatory control processes we normally understand the immune system to adopt, these Tregs, for example, T regulatory cells, those are not significantly different between the Amish and the Hutterites, right? They have exactly the same levels. So there's something else going on. When we look at the transcriptional profile of those leukocytes, what we see is that the infants in the Amish community have a massive upregulation in the transcription of two key factors. You can't really see that on this thing, but TNF up there in the middle, that's tumor necrosis factor, and IRF7, so that's a, check it, interferon regulatory factor 7. I have been up since 4 a.m. See, I don't even know what time zone I'm in. And I think the altitude up here is affecting me. FYI. If I make more mistakes, that's the reason. Those two factors are involved in the innate immune response. The innate immune response to responding to how much microbial exposure those children are getting. Okay, great, right? So the Amish kids have this upregulation in microbial sensing in their immune response, suggesting they're getting more microbial exposure. And they have a reduction in the inflammatory potential in their blood system, right? So we think that those two factors combined are allowing their bodies to respond more appropriately to any allergen that comes into their exposure system. How do we prove that? Best way to prove it is animal models. So we developed with our colleagues at Arizona an animal model of hyper-airway responsiveness, hyper-responsiveness in the airway, and increased eosinophilia. Oh, that one, sorry. It increases eosinophilia. And the increased eosinophilia in these animal models generates a pattern whereby if we sensitize the animal with evalbumin, and then we give it evalbumin later, then the evalbumin will trigger this inflammatory response, this asthmatic-like attack. To see if we could figure out what was allowing the Amish kids to maintain protection against asthma, we developed another mechanism where we took dust from the bedrooms of those children. So we took dust from the bedrooms of the Amish kids, dust from the bedrooms of the Hutterite children, and then we blew the dust into the nasal passages of the mice, right? The mice, these are sensitized mice, remember? If I give them evalbumin, they're going to have an allergic reaction. When I blow the dust from the Hutterite community bedrooms, Hutterite children's bedrooms into the respiratory airway of these mice, it's not protective, okay? It's just dust. It doesn't help them at all. But the Amish dust is highly protective. Dust from the bedrooms of the Amish kids protects these animals against an allergic reaction to which they've already been sensitized. So somehow it's allowing their immune system to respond appropriately to that stimulus. So we're following this up now with a much larger study. The microbiome component of this original study published last year in New England Journal of Medicine was very small, and now suddenly it's like, oh my God, the microbes could be really important. So the next phase of this experiment is much, much bigger. And we're now embracing this and trying to understand the actual implications of the microbes in the dust and how it works. The most interesting thing is, right, not all Hutterite kids get asthma. When we go into the homes of Hutterite kids that develop asthma, the dust from those homes is not protective. The Hutterite kids that do not develop asthma, the dust in those homes is protective. So when you talk about built environment probiotics, if we could just grab dust from Amish homes and from the homes of Hutterite kids and then who don't develop asthma and then spread it into all the homes in the US, maybe we could help to reduce asthma rates overall. And potentially atopy as well, because a lot of the Amish kids have lower atopy rates. So we're really excited about this because it has real potential to have a translational impact upon our ability to understand these disease states. So this leads us to understanding the microbiome of the built environment, where these bacteria come from. We know, as and Paula pointed this out, work from many of our colleagues has demonstrated some really interesting numbers, like 38 million cells of bacteria released from your body every hour in your little microbial cloud, your little pig pen. You know, from the Peanuts cartoon, you're surrounded by these microbes. If I took a swab and I swabbed each of your chairs right now, I'd be able to identify that it was you sitting on that chair because you're leaving a microbial signature. Yeah, I did just pat my bottom, pardon me. But you're leaving a microbial signature even through your pants. Again, the steaming isn't helping. Right? And that microbial signature is there, and it's allowing us to forensically identify you because of that unique profile that you have. Okay? And so this microbiome in the built environment package, while it seems like a dead ecosystem that we can't glean any information from, is actually helping us to understand some really important points about what it is that allows children to survive. One of the key things is we can work with architects and building scientists in this system to try and design better buildings. So we're really excited about that. Work that we published back in 2014 was one of the first studies to really identify the longitudinal constraints of how microbes changed when the built environment changed. So for example, here we have 7 families. These are samples of microbial communities. Every dot is a microbial community. The closer the dots are together, the more similar the communities are. If we color these dots by person, home, or pet, you can see very different community structures. The pets have their own unique microbiome up there in the top left in red. And then the home and the people are quite overlapped, right? There's not a huge differentiation between them because the people are donating the vast majority of the microbiome into that environment. So the home environment is the human microbiome. And we found that we could model this using temporal dynamic modeling, Bayesian statistics. We could identify closest probability estimates of interactions between, say, a hand and a doorknob, or a foot and a floor. And it's hard to see, but this is actually my family. Don't tell the IRB that I told you that. Keep quiet, don't tell them. But here on the bottom left, this hand 1, that's the hand of my youngest son, and he was crawling at the time, right, and interacting with the floor a lot more, and he has a much bigger microbial interaction with the bedroom floor because he was the one physically interacting with that floor. When we add dogs into that environment, we see that dogs actually actively increase the degree of microbial sharing between people, opening up new avenues for microbial sharing. And work from some of our colleagues down in San Diego has demonstrated that couples that have a dog are more microbially similar overall, gastrointestinally and on their skin, than couples without a dog. So a dog helps to increase your microbial similarity, right? This is fascinating. Interestingly, it increases your microbial similarity even more than a baby does, right? So you think a baby brings you together, well, a dog does it more, okay? So you can't go and rescue a dog. Failing marriage material is perfect. And it gets you out and it gets you walking, whereas babies have you stuck inside trying to clean up poop. Dogs, you clean up poop outside or just leave it. One of the interesting things from that study was that we identified that we could see people's microbial signatures distributed differentially around the house. So these plots, each one of them is what we call a source tracking analysis. The x-axis is time, right? Daily sampling over a 6-week period. And the y-axis is the amount of bacteria, the percentage of bacteria that were found on that surface that came from one of these 6 sources. So you look at the kitchen counter, right? You have 3 people living in this house and 3 dogs. The vast majority of bacteria on the kitchen counter come from person 3, right? Person 1 is a 25-year-old man still living at home with his mum and dad, okay? Because why not? Person 2 is a 57-year-old man living at home with his 25-year-old son, okay? This is the dad. Person 3 is the 6-year-old mother. Okay? And the mother's microbiome is the predominant source on the kitchen counter. I mean, seriously? The gender inequality in this home is inappropriate at the best of times. But it's even worse when you see that when she goes away for a few days and her microbiome signature dissipates, the dad's microbiome signature explodes onto the kitchen counter as he desperately fends for himself in this new environment. The bedroom and the kitchen floors are predominantly dog bacteria, and we can map and track that and see which dogs are interacting with which surfaces more. And you have this weird peak here for the young man at 25 on the bedroom floor. I do not know what's happening there, and I do not want to know. But it's interesting, right? This means that we can identify what people are doing in that space. This led us, there's our current president on the front cover of Time magazine who covered this research. I won't go into that. But this led us to explore how the microbiome could be used in forensics. And so we now work with the Department of Justice, the National Institutes of Justice's research wing, to identify whether the microbiome could be used for forensic identification of a home invasion, okay? And this seems crazy, and it quite potentially possibly is, but it's actually based out of a lot of research that we've done, and we're now actively going around the country. This guy here is Jared Hampton Marcel, who leads this program in my lab. He's a South Chicago born and bred kid who's really interested in microbiome and is now becoming one of the world leaders in this area. We actively work with police forces and forensic teams around the US to try and identify microbial signatures when we have a break-in. We haven't actually done any real break-ins yet because we definitely know that this technology is not ready for prime time. There's no way we could use it to identify an actual perpetrator. This is pure research, right? But you've got to start somewhere. Most interesting thing about this was, these are source tracking plots, so it's a little bit complicated. But number one, there are multiple rooms in this house that we burgled. Bedroom, dance room, dining room, kitchen, living room. Yes, there is a dance room. This is Florida, after all, right? Every home in Florida has a dance room. And what we did is after the burglary, because we had the microbial signatures of the homeowners and of the burglars, we determined whether you could identify the signatures, the specific bacterial taxa that were present in that home environment, in that particular room, that came from one of these individuals. So most interestingly, the bedroom had a higher level of bacterial signature coming from burglar 1. The dance room had more bacteria come from burglar 2, so he obviously was a little bit more funky. Dining room is burglar 1, kitchen is burglar 1, living room's about the same, office was about the same, outside entry was predominantly burglar 1, and the personal item was burglar 1. We didn't know which burglar had actually interacted with which room to begin with, but then we followed up and we basically, they told us where they went, they told us who stole the personal item, which was an iPad, right? And then we basically went back in and said, well, absolutely, we can predict that. This is an N of 1. We've now done an N of 30. We've burglarized 30 homes in Florida, in New York, and in Chicago, because why not? You can. And this is Jared's team down here. We work with disadvantaged kids from South Chicago to engage them in science. It's a little bit odd. I live in a very, Chicago is very segregated. I work in a very, very white neighborhood, Naperville, in the western suburbs, and I actually bring these kids from South Chicago to come in and burglarize my friends' homes as part of science, right? So we have had the police come round, no joke, because there's a load of kids standing around outside a house with clipboards, and they're like, what's going on? Either way, it's pretty exciting. So Jared and his team have collected hundreds of samples with our collaborator Joe Lopez down in Fort Lauderdale as well. And we're now trying to start to disentangle this relationship and identify whether we can actually routinely determine the burglar's signature when they come into a home. Each of the signatures is based on 16S ribosomal RNA sequencing and metagenomics. These are 16S ribosomal RNA signatures using what we call a DBLUR, or another software package is called DAR2. It's a software package which allows us to identify unique sequences for every single one of the OTUs in our analysis. So you get a unique microbiome signature for each of the subspecies. We call them sub-OTUs, right? Because an operational taxonomic unit was the standard preparation. You cluster all of your sequences at 97% nucleotide identity and you hope for the best. But it's a cloud. That's a cloud of taxonomy. We wanted something more specific. We get these specific signatures. We take the burglar's signatures, we remove anything that's common between all burglars, we remove anything that's common between the burglars and the homeowners, and anything that's common between the burglars and the home prior to burglary, okay? We're weighting our analysis as best we can. Whatever's left over in the burglar's signature, we identify, we determine whether it can be used to determine if that person invaded the home post-burglary, because none of these burglars had ever been in these homes before. And indeed, the burglars and the homeowners each have unique microbial sequences which are specific to them, and interestingly are robust over the 2 years of the study that we've currently been doing. So each of the burglars has this robust signature, which we absolutely love. We don't know if it's robust for longer. We're working on that, right? Give me 10 years, 20 years, I'll keep on telling you. It's really hard to do a longitudinal time series of an individual person over a 20-year period when we've only really had the technology for 5 years to do it. But we're working on that, right? And we identify these unique signatures on door handles, in toilets, on the floors, and we try and see if the burglar broke into the home. Just to swap tasks a little bit, but in a very similar vein, this is our brand new hospital we opened in 2013 at University of Chicago. And in this, we did a very similar thing. We looked at the hospital environment and the patients and the doctors for 365 consecutive days, looking at, say, the nursing station area, the air and the water, the bedroom, the patient rooms, so the bed rail and the floor and the walls, etc. And then when the patients and the doctors moved in, about 2 months after we started the study, we kept on monitoring them. Remember, daily sampling. We lost one postdoc doing this study who went crazy and had to move to Texas. As you know, where's my Texas people? I can't see you. There you are, there's the Texas people. You know what it's like, right? So these are floor samples. The floor microbiome before and afterwards is significantly differentiated, and this is because prior to move-in, we didn't have a break in here, sorry, prior to the hospital becoming a functionally operational hospital environment, there were very few bacteria in the environment. Most of them were distributed from other sources, and so the blue dots are the microbiome before the move-in, and then the red dots are the microbiome of the floors of the patient rooms after the hospital became operational, and they are saturated with human-associated bacteria. And that's great. We found in this study a lot of similarities to all of the other studies, that humans deposit their bacteria into the environment when they move in. The patient rooms were identifiable to the patient, right? I could swab the floor and identify which patient was in that room. But we could also do things like track the antimicrobial resistance, because we also did shotgun metagenomics on a large number of these samples. And the most interesting, the antibiotic administration was mostly intravenous or oral, but we did have some skin-associated antibiotic flora, and those are in brown here. All of these samples are samples from when the patient was taking antibiotics, and these are from post-antibiotic samples, right? So you get a differentiation. Post-antibiotics, the microbial diversity rebounded and stayed quite routine across all the patients. During antibiotic treatment, there was high variability in whether an antibiotic had an impact. Intravenous antibiotics have very little impact upon the skin bacteria. Skin-associated antibiotics, like neomycin, had a massive impact upon skin bacteria. Okay, great. So these are the microbial stressors, okay, and what's affecting the microbial community in that environment. We then went and checked out where the antibiotic resistance genes resided. Tetracycline resistance genes were the most common on skin, probably to be expected, because we normally do tetracycline administration on skin. And then we have multi-drug efflux proteins being more abundant on surfaces. The skin antibiotic resistance genes were predominantly in skin bacteria, Staphylococcus aureus, Staph epidermis, Corynebacterium striatum, whereas the ones on surfaces predominantly came from E. coli and Pseudomonas aeruginosa. So these are differences, right? The antibiotic resistance profiles of this environment is differentiated across the people and the hospital environment. And when we dig down further into this and we start to reconstruct the genomes of those organisms and look at the distribution of antibiotic resistance genes across those systems, we can see two interesting things. Number one, we're able to get these organisms like Staph epidermis, Propylacterium acnes, and Enterococcus. We reassembled their genomes, and we see that they're stable over time in the environment. But most interestingly, we have the same genome 70 days apart. And 70 days apart, over that time frame, it's actually started to acquire antibiotic resistance in the hospital environment. This did not happen before the hospital was operational. It only started, the acquisition of antibiotic resistance genes by these organisms only started once the hospital became a functionally operational environment, okay? So the impact of actually using this environment for medical practice is having a defined evolutionary selection pressure on the bacteria living in that ecosystem. And we want to understand this much more significantly, obviously. I'm not going to go into this. This is Miles. Miles is sitting there, the hand raising. Miles is getting a poster on this, but we started a dorm study just prior to the one we're now doing with Andrew, right? Lieutenant Colonel, sorry, to try and understand the impact of microbes on dorms. We've got students our students are much less rigorously controlled than the cadets. If you've ever been to a university campus, and many of you have, you'll understand what I say. Interestingly, the microbial community is much more similar between surfaces that interact, as we know. So over here we have shoe samples in blue and the floor samples in green. So the shoe and the floor match up because the people are interacting, the shoes and the floor are interacting. And then over on the left it's hands, desks, bed sheets, and door handles over here, all clustering together because you're sharing microbes between those hands. And that's obviously at least you're standing outside the boys' toilets there, but you're swabbing the toilet door handle. I love this. You know, this is your natural territory. But this map down here is a map of each of the interactions between the people, the students that were actually engaging in the study, right? So you can actually start to see beta diversity associations between the bacterial communities that provide similarity. So you can see which people in the dorm environment are physically or microbially interacting. They don't necessarily have to be touching, but they may be, which obviously leads to some ethical complications when you actually divulge that information, but we have full IRB approval and we're sworn to secrecy about who these people are. But this is great, so we can track social interactions, and I strongly suggest going and having a chat with Miles, because he knows this study much better than I do. Just to quickly finish up, I just wanted to highlight one more study that we started last year, which is an investigation of the microbial metabolism. We wanted to know, if I put my hand down on this surface and I leave bacteria and fungi there, when I take my hand away, what happens? And now Noah Fiera is going to be here tomorrow, and Noah's entire research paradigm is essentially they all die. And some of them do. But I'd say about 99% of them do. But some of them don't. And if you accidentally spill your coffee on there, or some water, or you have a flood, then the microbiome in that environment is suddenly going to become alive, metabolically alive, and it's going to change. And when we speak to our built environment colleagues, the one thing they're interested in is what happens to the built environment when it gets wet, right? That's the key thing, because then the fungi grow, and then you have the health problems. And so what we wanted to understand is we wanted to apply all of our tools and techniques to metabolically track this environment. So we want to understand how the microorganisms, mostly the fungi and bacteria, metabolically interact on these surface materials, right? We basically take inoculants. We want to determine the inoculant impacts growth rates of the fungi after wetting. We want to know if metabolic interactions shape those growth rates, so if the bacteria either are detrimental or advantageous to the growth of the fungi. And then we want to understand if the surface characteristics, if the type of material I'm touching or interacting with shapes the growth potential of the organisms in that environment. So this is Dan. She works in Brett Stevens, thank you, lab in the Illinois Institute of Technology. And these are all our incubators. We do sterile inoculation of surface coupons of wood, or plywood, etc. And then we either wet them or dry them. And these are some of the coupons with fungi growing on them after a period of time. And we do multiple things to this data set. Number one, we can look at the abundance of the organisms, right? So over here on the left is work from San Diego State University, Scott Kelly. I'm really bad with names. I need a name dictionary in front of me. Where we actually do fluorescent microscopy to identify the density of organisms growing on the surface. The top is a surface that hasn't been wetted, and the bottom is one that has been wetted. You can immediately see growth. And then we apply amplicon sequencing, metagenomics, metatranscriptomics, metabolomics, fluorescent microscopy, as I said, qPCR, temperature, humidity, CO2 respiration rates, chemical surface analysis, and surface structure. And then we basically use systems biology and systems ecology thinking to try and drive all of this data together and come out with an understanding of how this organism metabolically interacts with that organism and leads to either the increase or decrease in its growth rate. And surface respiration. We know, for example, the bacteria in archaea show very big, strong differentiations based on moisture. So moisture here is in circles, and wet is in triangles. So the triangles are all over on the left. Microbial community in a wet environment is very different from a dry environment. That's great. And to a lesser extent, material. So gypsum, medium density fiberboard in blue, Green is a mold-resistant type of gypsum, and then the yellow is plywood. And there is some differentiation. You get some clustering of the community profiles of the bacteria and archaea, but based on that. If we look at the metabolites, I'll show you the fungi in a minute, but look at the metabolites, we see an interestingly different pattern. Clear clustering up at the top for metabolite profiles of these systems based on dry or wet. Dry, not much activity; wet, huge activity. Very clear differentiation based on the material. The material these organisms are growing on shapes their metabolism, because it shapes their labile carbon sources, it shapes their nitrogen sources, shapes the kind of thing they can access. And then where we get the inoculum from, this is from a home with children, this is from a home without children and with a dog, for example, shapes the types of microbial interactions and metabolic processes which go on that surface. Not necessarily the organisms that are present, although that does have an effect, but definitely the metabolic relationships, and we don't know why. We can map all of these. These are the 60ness and ITS. So ITS is for fungal organisms and correlations. The big squares are fungal taxa. The round circles are bacterial taxa. And you can start to, using a random walk Bayesian classification mechanism, you can start to identify clusters within these network associations. So these are Spearman rank network associations with only very highly FDR approved, FDR validated significances. And you get these different groups. So we have a group 1 is blue, group 4 is green, group 3 is light green, group 5 is pink, and group 2 is blue. And then we can see where these groups are most enriched. So up at the top, I've colored it by dry enriched or wet enriched. So those groups 1 and 4 are heavily enriched in the wet environment, as is part of this big group 3 down here, right? So you get that differential enrichment. Location enrichment is spread throughout the system, but we can see different locations affect the partnerships and associations between the fungi and bacteria. And then we can determine how the microbial community is responding in terms of its growth function, or its differential relative proportions and associations across the different materials. So for example, this is for group 1. Group 1 has a very strong positive correlation with medium density fiberboard and wetness. So we see that association become very abundant of those fungi and those bacteria coming together and metabolically interacting when you put water onto the medium, onto the MDF, onto the mold-resistant gypsum. And then the metabolite network here, done exactly the same. This is just for metabolites. But we said, what's enriched in the wet environment, what's enriched in the dry environment, and how is it shaped by where the samples come from. And then we, again, do the co-association integration mapping. So for example, here, this cluster of metabolites over here, number 2, is heavily correlated with the material plywood and less correlated with wetness, but heavily associated with other environments. So these interaction maps allow us to hone in on exactly what's going on in terms of the associations between bacteria, fungi, and metabolites. If we interact all of those together, this is the last couple of slides, so I'll stop in a second. And these are bacteria, fungi, and metabolites all co-interacting, only highly significant interactions. We can start to see partnerships forming and the bridges between those partnerships. So why does this cluster of bacteria, fungi, and metabolites interact with this cluster of bacteria, fungi, and metabolites? And what are the partnerships which drive that? So currently, the connections between the nodes of the same type are not included here, so we're only looking for connections between either bacteria and fungi and bacteria, or fungi and metabolites, bacteria and metabolites. And that shapes how we can partner up these. So for example, that little green guy, I can't point to it, but that little green guy right up there in the middle, that little green metabolite, and that little bacteria, that's acetate, right? And acetate is shaping the interaction between the module of interactions at the top and the module of interactions down here. There's a carbon constraint upon the relationship between those two components, and the organism is an Aerococcus, okay? So common skin-associated bacteria. So we see that relationship starting to build. We are going over this with a fine-tooth comb and now using it to define new experiments. The one thing we're doing right now is we're identifying all of these super important metabolite compounds. So we have about 24 of these. Each one has 25 or more connections in this network. And we're using that to try and say, what are they, why are they important, and how could they be shaping the interaction space? So We're currently defining the consortia, using defined consortia of common built environment fungi and bacteria to create definitive co-associations, and we're mapping the metabolic relationships and predicting respiration rates based on those relationships in silico, and then refining the prediction of the growth outcomes of these key fungi and bacteria. So we're literally taking that environmental constraint, what happens when you wet the gypsum and it becomes fungally overgrown. We're deriving that and trying to put it into a computer and using that computer algorithm to predict outcomes in that system. Why are we doing that? We want to identify the specific elements which retard fungal growth. Because we think if we can do that, we can prevent fungal growth in homes, potentially putting materials into gypsum or into plywood or into plasterboard that affect the growth potential in that environment. We can control it more effectively. So we have a microbiome center at University of Chicago, our national lab, combining expertise across all the different groups. And I have a book coming out on June 6th, shameless self-promotion. If you want to know more about, if you have a child coming or a grandchild, or you're planning for kids, this is the book for you. It's What to Expect When You're Expecting for the Microbiome. It's written by myself and my colleague Rob Knight and Sandra Blakesley. And it's called Dirt is Good. Thank you very much.